Okay, so we're going to take a look at the cerebellum. I have noticed that not all of you have taken advantage of watching the other videos, so we're going to have a pop quiz later this week on the material that you've been presented with so far. I'll take a look at it, I'll let you know so you have time to get caught up. So I will only post one lecture this week, but I want you to take the time during our regular class time to watch these lectures. It does me no good to make them if you're not going to look at them. And unfortunately, the material is far too complex for you to try to do this in one night. So do yourself a favor and keep up because it's easy to just procrastinate, and that will be really not great for any of us. All right, so let's talk about the cerebellum. It's a pretty interesting area of the brain. In fact, there are many scientists who only study the cerebellum. It only is about 10% of the total brain volume, but it has way more neurons than the rest of the brain. So it's a pretty interesting area to look at. It's really not very well known, as you've probably heard me say a lot, but we know less about the cerebellum than we do other brain regions, but we do understand some basics about it. You've heard and been taught that control of movement is really what the cerebellum is about. But it doesn't have any direct connections with spinal motor neurons or brainstem motor nuclei. So there are no direct connections. All of the connections are indirect. They've also found that it does have a function in cognition and learning, motor learning particularly. And so we can kind of tell that if somebody has damage to the cerebellum, and then they do have difficulty learning motor tasks or other, cognitive tasks. So unfortunately, sometimes we find out what a brain area does when people are injured in that area. It's one of the only ways we've been able to, prior to a lot of the brain scans, we've been able to tell a lot about the function of a particular area of the brain. So we'll see that the microcircuitry of the cerebellum is really cool, actually. It's highly regular repeating units, and it's basically the same microcircuit that's repeated, obviously, in abundance since it has more neurons than the entire rest of the brain. Also, we see that different regions of the cerebellum do get inputs from different areas. So, and it has a lot to do with organization. We see that it is somatotopically organized, similar to the motor cortex and the SMA, that different regions are related to different areas. And it kind of makes sense because in the cerebellum, the more central regions, the more medial regions, control the more medial parts of the body. And as you go outward, if you get more distal, then the areas there, the association areas, control more of the limbs and the more distal areas of the body. So at least it makes sense, right? I'm not going to ask you to learn all of the different areas and nuclei. Something I had to do. I think it's really interesting. I think it's beyond the scope of what we will try to accomplish this semester. But there are a few things that you will need to know. Internal models that, and it says discussed previously, we haven't really gone into a lot of internal modeling regarding motor control. So don't worry so much about that terminology. These are from Dr. Poston's lectures. But we do see that there's some belief now, some evidence that shows that internal representations of the external world are, partially implemented in the cerebellum. So not only does it have some implications for movement and motor learning, but also cognition and awareness. So here we see, believe it or not, this is a fairly simplified version of some of this anatomy. It's quite complex, but we see that we have this outer mantle of gray matter. And so remember what gray matter is, and that's usually the cell bodies. It's non-myelinated. And so then we see that then the white matter is within, similar to the arrangement of the cerebral cortex. And so remember what white matter is. It's going to be myelinated. And what is the cell that myelinates the neurons in the central nervous system? That might be a good question for you to consider. So, Schwann cells take care of the peripheral nervous system. So that means that oligodendrocytes myelinate the neurons in the central nervous system. And then there are 3 pair of deep nuclei. And we see here that we have the vestigial, the dentate, and the interposed nuclei. And there are 2 in the interpose, the emboliform and the globose. So again, I don't know that I'm going to ask you to memorize all of the functions of those, but I need you to take a look at where they are located. So we say the vestigial is more medial, and then the globose nucleus and the emboliform nucleus would be central between the other 2, and the dentate nucleus, which is much larger, is more distal. There we go. So also we can see that the peduncles, you remember the cerebral peduncles from your anatomy class, and you always kind of wondered what those are for? Well, they're pretty important when we're talking about cerebellar anatomy, because they connect to the dorsal aspect of the brainstem through these peduncles. And so remember what those looked like when you did your dissections. Now all of these structures do have very specific functions, and we'll talk about some of them. But we do have some basic pathways, both in and out, since there are no direct connections. Obviously, the communication is going to have to travel via efferent and afferent pathways. And so they travel through these 3 major fiber bundles, and those are your peduncles. So those peduncles are the fiber bundles that transmit the information back and forth from the cerebellum to the rest of the structures. And there are 40 times more fibers that enter than leave the cerebellum. So what does this indicate? We've got a lot of information coming in, a lot of processing happening, and then directions based, right? Your instructional information coming through your efferent pathway. So that's quite a lot if you think of it. I think that explains a lot of the function generally of the cerebellum, and it's certainly its importance. So the outputs travel through the superior peduncle, and the inputs in the middle and inferior cerebellar peduncles. Most of the afferents come from the pontine through the middle cerebral peduncle, and the rest come through the olive or the spinocerebellar tracts from the spinal cord. And I always wondered what the olive was for. I thought that was kind of a cool structure in the brain when I was learning anatomy. So now I know it has some involvement here. All right, so we're going to talk a bit about spinocerebellar tracts, and we've got a picture coming up. I'll show it to you here. There we go. So you've got this in your slides. So we've got these tracts that send the information to the cerebellum, and specifically all of those sensory organs that we've talked about ad nauseum earlier in the semester, as well as from skin receptors that you learned more about in anatomy and physiology. And we have a posterior spinocerebellar tract getting sensory information from the lower limb, and the cuneo-cerebellar tract, the upper limb, the anterior, more proprioception from the lower limb, and the rostral from the upper limb with descending input. So these are the different tracts that we relate to the cerebellum. So here's a nice outline for you. I think it's a good idea to have an understanding that there are different tracts that receive and also transmit the afferent and efferent pathways. At this point, I don't believe I'll be asking you a lot of questions. I would like for you to pay attention to the basics though. So posterior and anterior, there's really only 4. I'm trying to be nice to you, but at some point you're going to have to hold you accountable for some of this information. You might expect to see a question, a multiple choice question on this in a general fashion, and you might be aware of these different tracts. This is a nice study outline for you. You can take a look at this in more detail. I'll be sure to post this lecture for you also in full slides, so you have some of these diagrams in a larger format. All right, so we do see that we have different regions, functional regions in the cerebellum. And again, I want you to have a general idea. If you take a look at this, it's a pretty fascinating diagram. So the vestibulocerebellum, you take those two words, you have a pretty good idea. So we're thinking of the vestibular system, right? We're thinking about balance often, eye movements. You might remember your cranial nerves from A and P, and I think it's cranial nerve number 8 that has to do with your vestibular system. I think it's your vestibular coccylar nerve, and so this is also part of this system. But it does consist also of this flocculonodular lobe, and it is something that sometimes if it is damaged or disrupted, then we see a real distinct loss of balance. And so you may see that in the literature sometimes in relation to balance. It is the most primitive part, and it does receive vestibular, so balance and visual input. So you can see that damage to that part of the cerebellum would be pretty incompatible with successful life. The spinocerebellum, and here we have the vermis and the intermediate parts of the hemispheres. And so we're going to find more proprioceptive inputs from the spinal cord. So this is where we're going to start getting that sensation. And so the vermis, which you hopefully remember or familiar with, it receives lots of information from the somatary system input from the head and the proximal body parts. It projects via the vestigial nucleus to the cortical and brainstem, and that helps to control proximal muscles. So here we're inward and we're moving outward, you see. So now we're controlling proximal muscles. We're controlling more things that are closer to the body. So again, the vermis is very important in locomotion, posture, and eye movements. And we have some neurons that project there into other areas of the brain and the cortical spinal and rubral spinal systems. And we'll be talking about what those are. And then the cerebral cerebellum. So what do you suppose that is? We have this connection from the cerebrum to the cerebellum, and that is on the outside. That is the farthest, most distal, and it's so the most lateral. The inputs and outputs are going to the various cortices of the cortex. And so we see some inputs from the association cortex. We see outputs going through the dentate nucleus to premotor, motor, and prefrontal cortices. So this is also very dedicated to other areas, and I like the way that they've drawn out this diagram. You can see from the arrows what areas of the brain we have inputs and outputs from and to. As far as detail goes, I would expect that if I asked you a question about general function of the spinocerebellum or the cerebrocerebellum region of this organ, that you would have a general idea of what it controls. Let's take a look a little bit at the microcircuitry, which I think is incredibly fascinating. So neurons are organized in 3 distinctly different layers, and this is uniform throughout. Remember in our first slide we said that we have a lot of repeated units. It's pretty regulated. All right, so the granule layer, the 100 billion granule cells, and a few Golgi interneurons that inhibit the granule cells. So the reason we have these Golgi interneurons is largely inhibitory. In fact, most of the activity we see in the cerebellum is inhibitory. So we see a lot of GABA, so a lot of inhibitory as opposed to excitatory circuits. So the mossy fibers terminate here. We're going to pay attention to two distinct types of fibers: mossy fibers and climbing fibers. So mossy fibers are bringing they're afferent fibers, so they're bringing stuff into the cerebellum, and they terminate right here. Um, and we see in our book, Latache talks about the glomeruli, the cerebellar glomeruli, and basically that is where the cells from the granular layer make synaptic contacts with the bulbous expansions of the afferent mossy fibers. A single glomerulus consists of an incoming mossy fiber, clusters of small dendrites called rosettes from a few dozen granule cells, and axons of Golgi cells. So a single mossy fiber may innervate many glomeruli. Also, we have a layer of Purkinje cells, so our second layer are Purkinje cells. And this is a single sheet, so that's that center right there. It is an output layer. And we see that these dendrites from the Purkinje cells extend into the molecular layer. And that's where they're getting inputs from climbing fibers. That's that second major type, okay? So dendrites are getting inputs from climbing fibers. And they also get some excitatory and inhibitory interneurons in that one, input from those types of interneurons. So we see some excitatory there. Again, most of what we're seeing here is going to be inhibitory. Purkinje cell axons are really responsible for conducting all of the output. So anything in the cerebellum has to go through the Purkinje cells. So if we have issues with Purkinje cells, we obviously have issue with any type of involvement in the cerebellum. So you see that they project deep into the deep nuclei of the white matter, and they have an inhibitory effect. So Purkinje cells, in this case, are going to have an inhibitory effect. And then the molecular layer is the processing layer, and that's that gray matter that we see, that we're fond of seeing. The axons of the granule cells are referred to as parallel fibers, and they run perpendicular to Purkinje cells. So they're really kind of an interesting way this works. So each granule cell can have lots of synapses with a large number of Purkinje cells, and this is, not only do we have more circuitry, not only do we have more neurons, but they also have massive connections. So you can kind of see in the drawing there how we have these cells that are running parallel and perpendicular, and so all of this allows continual communication between all of the neurons, which, if you're thinking of information processing, is incredibly powerful. So mossy and climbing fibers are the two afferents, and they both form excitatory synapses, but they terminate in different layers, and they produce different patterns of firing and mediate different functions. So they are, although I've told you most of the, so I was going to say most of the output is inhibitory. I don't want to go back and re-record all that. So we do see excitatory synapses, but we see most of the output work of the Purkinje cells, which is where we get the output, that is inhibitory. Okay, so we excite them, and the response is inhibitory. Speaking specifically to mossy fibers, they come from the cell bodies in the spinal cord, and they are transmitting lots of sensory information, and also from the cortex. So they're going to produce these firing, referred to as simple spikes. And I think our book goes into simple spikes and complex spikes. So this is in the Purkinje cell. And each granule cell is going to receive input from only a few mossy fibers, but each granule cell is going to actually distribute the information to a huge number of Purkinje cells through the parallel fibers. So it's almost like telephone wires, right? Kind of think of these parallel fibers running across all these Purkinje cells. We see the mossy fibers making contact with the granule cells, taking that information up to the telephone wires. And if you think of this arrangement, you can understand and appreciate the complexity of the cerebellum and why perhaps we don't know as much about it as we like. So each Purkinje cell is contacted by the axons of between 200,000 and 1 million granule cells. So this is a pretty rich and robust arrangement. And then the climbing fibers, they are originating in inferior olivary nucleus. And they're bringing in sensory information from the periphery and the cerebral cortex. And they produce firing as known as complex spikes. So we see that mossy fibers produce simple spikes, climbing fibers produce complex spikes. And they run parallel to the Purkinje cell. And again, this is going to allow them maximal exposure to the information stream, to the telephone wires, if you will. So we see that each Purkinje neuron receives input from only one climbing fiber. So it's only one climbing fiber per Purkinje cell, but still, because of this unique and complex arrangement, it's high-level processing. So they have the different effects on Purkinje cells. Remember, mossy has the simple spike, and climbing has the complex spike, and these have different actions, different repercussions. Climbing fibers have a strong effect. Makes sense if it's more complex. We see a greater response, and an increase in calcium conductance. And so that means a longer depolarization and involves a large amplitude followed by some high frequency bursts. And your book has a nice picture of that on page 155. So when stimulated, these climbing fibers fire single action potentials, and they have a specific timing, so that timing can be measured. And if we can get an idea of what a healthy timing would look like, then we can begin to understand some of the underlying issues in movement disorders that are related to the cerebellum. We see that the synchronous firing of many climbing fibers enables them to single important events. So it's not necessarily correlated to behavioral measures, but more in event detection. So yeah, synchrony is a really interesting topic when we talk about the brain. And we like to think of things to be cause and effect, right? This happens, and so this happens, and then this happens. And our brain and our science, this very didactic kind of process-oriented way of learning and teaching, again, we kind of get hypnotized into the belief that everything works that way. And in fact, it's probably more likely that the brain produces synchronous results and also is affected by the synchronous firing, right? So we see this in EEG measures. We see that EEG measures of certain bands of electricity, of activity in the brain, if you will, is associated with certain behaviors and mindsets and moods and states of alertness, and also seems to have an effect on those very things. So it's something that the system is doing, and also at the same time is being affected by its own synchronous activity. So anyway, I think that it's just something to keep in mind, that the synchronous firing is more important than it might look on first blush. Parallel fibers, mostly dependent on mossy fiber input, is short-lived excitatory potentials, and so single parallel fiber is very weak, but again, we look at the culmination of them. So a large number of parallel fibers are needed to have a big result, because we have this simple spike. And also, the Purkinje cells have spontaneous firing rates, 100 spikes per second, even at rest. There's a lot going on. All this information is coming in. And of course, it must process it, because remember, it has 40 times more inputs than outputs. And they believe the mossy fiber system, there's evidence that shows that it encodes the magnitude and duration of peripheral stimuli or central generated behaviors. And those are often thought of like walking and balance and some of the more gross movements that can't really truly be explained by reflexes and yet are still operating at the level of the unconscious. There's also evidence from some of the spinalized animals in those experiments that you can remove these higher centers of the nervous system and still produce these central patterns. So another piece of evidence towards central pattern generators, when they can still produce walking-like movement from an animal on a treadmill even after the animal has been spinalized. So just briefly, some of the information about the role of the cerebellum in motor learning and in learning. So we see that there are studies that have shown that the cerebellum is involved in 3 different forms of learning. I sort of left out the slides about the gain modulation of reflex responses. I felt it was beyond what we needed to address. But we do see that reflexes, if you will, are strengthened within the cerebellum, according to some research. We will take a quick look at classical learning and Pavlovian conditioning tasks, and also make note of the research done in the recalibration of voluntary arm movements depending on the task conditions. All right. Also, your book talks about a role in the creation and storage of internal models of interaction between the body and the environment. So, again, just briefly, internal models are hypothetical neural processes that are attempting to compute either neural commands based on a required mechanical action or changes in the mechanical state of the body based on current neural signals and sensory information. So basically, internal models, they're computing both, what changes need to occur in response to the environment and what changes in the environment are already occurring that requires a response. So there's another hypothesis that the cerebellum plays a role in assembling motor synergies. And remember, the synergies are coherent movements, according to Latache, resulting from coordinated actions of the individual elements, such as limbs, joints, and muscles. So we see that there's a lot of debate and still a lot yet to learn about the cerebellum. Anyway, so just talking about this Pavlovian conditioning task. So if they put an air puff in a rabbit's eye, makes me kind of sad. Anyway, and it causes the third eyelid to close. They pair that with a sound, and then the rabbit is conditioned to have that same reflex, which is obviously unconscious, when they hear the sound alone. So rabbits and cats show they can maintain this same effect even after the hippocampus, thalamus, or cortex is removed. So this indicates that it is the cerebellum that learns this response. However, this learning doesn't occur if there are lesions during lesions, so they can give a lesion through a magnetic pulse or electrical pulse, or sometimes through medication or removal of the lateral cerebellum dentate or interpositus. So we see that here is some evidence that it is the cerebellum that is largely responsible for the Pavlovian learning. And then recalibration of arm movements. So they train monkeys to flex and extend to catch a moving target. And so then they change things so that the monkey has to adapt, recalibrate. And it can be done in 10 to 12 trials, but it is not accomplished if, in fact, there is a virtual lesion created in the cerebral hemisphere through this mucimol, something that basically numbs or produces a virtual lesion. So it says the injection only affects the rescaling, but not the ability to perform. So the animal can still perform the general movement, but the rescaling ability is gone when we have this virtual lesion on the cerebellum. From this slide, lots of good information here. We see that it is the relationship between the climbing fibers and the mossy fibers. through the Purkinje neurons. That is, well, that's where we see the areas of activity during learning. And so it is believed that that is the pathway for this type of learning. And we see that climbing fibers modulate the input of parallel fibers through long-term depression. So in your book, we have some information about long-term depression and long-term potentiation. So do take a look at those explanations on page 159 in your book. As I mentioned earlier, we have learned a lot about functions of the cerebellum, as we have about functions of different areas of the brain, through people who have had injuries or disease states that have affected those areas. And so this is valuable for neurologists if a patient comes in with some neurological complaints to be able to tell whether the cerebellum might be involved. So these 4 symptoms that are hallmarks of cerebellar dysfunction would be hypotonia, as you can read there. It's a diminished resistance to passive limb displacements. It's also thought of to be related to the so-called pendular reflex. So the leg normally comes to rest immediately after a knee jerk produced by a tendon tap, but patients who have cerebellar disease, the leg may oscillate like a pendulum as many as 8 times before coming to a rest. So, with that basic knee tap, that knee reflex, that can be a very valuable diagnostic tool for someone with a cerebellar dysfunction. The ataxia is the inability to stand or walk. Ataxia, loss of ability to maintain a steady limb or body posture across multiple joints. Abasia is the loss of ability to maintain upright stance against gravity. So when standing or sitting, many cerebellar patients compensate by spreading their feet and attempt to stabilize balance by increasing their base of support. They move their legs irregularly and they often fall. The third symptom is ataxia. It's the abnormal execution of multi-jointed voluntary movements. It's characterized by a lack of coordination. So patients who have problems initiating responses with the affected limb and controlling the size of the movement and the rate of the regularly repeated movements, this is described as ataxia. This last deficit, first described by Babinski, you may remember the Babinski reflex, is most readily demonstrated when a patient attempts to perform rapid alternating movements, such as alternately rubbing the back and the palm of one hand with the palm of the other. Patients cannot sustain a regular rhythm or produce an even amount of force. So, the scientist Holmes also noted that patients made errors in the timing of the components of complex multi-joint movements, so like walking, and frequently failed to brace proximal joints against the forces generated by the movement of more distal joints. So you can see that this would be quite a problem for walking. And then the fourth symptom is a form of tremor at the end of the movement when the patient attempts to stop. So the action or intention tremor is the result of a series of erroneous corrections of the movement. So the cerebellum is overcorrecting. Once a movement is clearly headed in the wrong direction, attempts to make corrections fail repeatedly and the hand oscillates irregularly around the target in a characteristic terminal tremor. This behavior clearly suggests that the cerebellum normally is responsible for the properly timed sequence of activation in agonist and antagonist muscles, and that the loss of proper timing causes movements that, although initiated in the correct direction, cannot be controlled or brought to an accurate endpoint. So one of the conspicuous features of cerebellar disorders is this loss of automatic, unconscious nature of most movements, especially for motor acts made of multiple sequential movements. So of one of Holmes' patients who had a lesion on his right cerebellar hemisphere, he reported that movements of my left arm are done subconsciously, but I have to think out each movement of the right arm. I can come to a dead stop in turning, and I have to think before I start again. And so normally movement is controlled seamlessly, by these cerebellar inputs and outputs. With a malfunctioning cerebellum, it seems that the cerebral cortex needs to play a more active role in programming the details of motor actions. So this is from the Big Blue Book. So we can kind of see here. I'll let you take a look at this on your own. Kind of goes over some of the things that I just went through. But we see these patterns, and the timing, etc., is not able to be maintained with those with cerebellar dysfunction. So some of the strongest evidence is also this timing that I mentioned between the agonist and antagonist. And you remember that triphasic burst that we looked at, that agonist, antagonist, and then the second agonist burst? Well, that's changed in a person with a cerebellar lesion. So this is another thing that can be measured to identify what area or areas of the brain and the cerebellum have been altered by, by an injury. And so they cool the deep nuclei, and it decreases and prolongs the agonist burst. So we see that it has a very distinct role in the agonist and antagonist relationship in movement. So again, we see the triphasic pattern, and here's just some more evidence that kind of shows you this. We also can see that terminal tremor at the end of the movement. So EMG can actually give you quite a lot of information. So we see that those with cerebellar ataxia cannot account for the perturbing effects of joint interaction torques during multi-joint arm movement. So they simply cannot adjust accordingly. They cannot make those reactionary changes. So this leads to impaired movement trajectory and endpoint accuracy control. Also, we see that the damaged cerebellum also has a direct effect on cognitive function, time, length of sound, speed of objects, and some of these other things that you can see listed. So these, again, probably related to motor actions that require a sequence of movements. So it seems that the cerebellum is largely involved in this sequential aspect of brain function. So an important point is that, yes, we have issues. Movement is affected. We no longer have smooth movements in some cases, but the gross movement is still there, where if we have a lesion in the cerebrum, then we're looking at paralysis, right? So if we're over a motor center in the cerebrum, we're going to have paralysis. If we're in the cerebellum, we're just going to have a lack of control of smooth movement. So this is along with the fact that these inputs and outputs, it indicates that it compares these internal feedback signals. That is kind of like that comparator, which you may have heard before. So it's taking what is desired and then comparing it with the outcome. So in short periods, making movements, the cerebellum can generate corrective signals. They are feed forward, they're anticipatory, and they act on the descending systems of the cortex and brainstem. But remember, they don't have any direct contact. So they all have to work through these pathways through the other layers of the brain. Climbing fibers activity likely signals movement errors. So remember, those climbing fibers have those complex spikes. There's more communication that's going on. There's one climbing fiber per Purkinje cell, and so they have a pretty direct effect on modulating movement errors. All right, so I've added these slides for you. These are provided by your book, really just because I'm encouraging you to read your book. I think it has this information and probably more, but it's not laid out too badly. I just wanted you to have a different lecture that maybe has some better graphics, and then you can go through the remaining slides on your own. You're held responsible basically for what I went through in the slides that I have recorded.